So, Yaeko.
Okay.
Press it when you're ready.
Okay, I pressed
Now
it now.
Well, you can press it whenever, and then we can
Oh,
just trim the
okay.
video.
Start recording. Okay. I'm gonna share my screen. Yay, can you guys see my screen? Okay, yeah. Okay. So, uh, I don't know if I press can you guys see it fine? Okay. Yay. So, hi, I'm Yaeko. This is my project I've been working on with Embry, but first I'm going to talk a little bit about myself. So I just graduated from Hilo this May with a BS in biology and a chemistry minor. And for all 4 of my years, I was in AMSA, and for 2 years I was an AMSA treasurer, and then I was the AMSA president last year. And here are all these pictures of us, and I really had a great time um, in AMSA. And for 3 of my 4 years, I was an Embry student. Embry is an undergraduate program that lets undergraduate students have biomedical research experience. And because of the Embry program, I was introduced, or they offered me a job at the USDA Agricultural Research Services. It's like across the street from the College of Pharmacy. And like right now, I'm studying for the MCAT, and I want to go to medical school and become a pathologist. Oh, and I'm from Hawaii, so I was born in Hawaii, but I was raised in Texas and Louisiana. But then I came back for high school and go Hilo High Vikings. Okay. Okay, so the Embry research program is federally funded by the NIH, and I think it's a great program if you're interested in MD and PhD programs. And for me, I got a lot of poster presentation experience. I don't really like, my least favorite is out of the research is presenting, and I got a whole lot of presenting experience from this. So I don't know if you can see my arrow, but the top picture, every year we go to JAPSOM and we present our research that we did in the previous semesters. And this was last year. And then on the bottom left picture, I was privileged, and I was picked to represent the state of Hawaii at the NIH Western Regional Conference in Las Vegas. And here, the bottom left picture is my poster, and I was super nervous. And then on the right lower picture, I got to present at the Rat Lungworm Conference in January, which AMSA we volunteered, which was awesome. And I found out the day before my presentation that I was the only undergraduate person presenting, which was, I was dying inside because everybody else had their PhD and MD. I mean, their PhD, the medical degree, and their masters, and I was like, what? But I think it was a great experience. And also, if you're pursuing research, Embry's like perfect for you if you want to do biomedical research after undergraduate. And then publications, I got to be on 2 publications or published scientific articles. The first one, I was a part of my sophomore year, so when I first started Embry. It's titled Water Transmission Potential of Angiostrongylus cantonensis, Larvae Viability and Effectiveness of Rainwater Catchment Sediment Filters. So we basically tested different size filters against this rat lungworm. And then my second publication is the demographics of the semi-slug Parmenian martensi, an intermediate host for Angiostrongylus cantonensis in Hawaii during laboratory rearing. So we reared semi-slugs for rat lungworm experiments. And I was like, the second publication I was more involved in than the first one. Okay, and so the project, my research that I'm going to be presenting today, I've been working on this for almost 3 years. It's the efficacy of electromagnetic irradiation of the parasitic nematode Angiostrongylus cantonensis. So specifically, we focused on UVC and X-ray radiation. So my co-authors or my instructor is Lisa Kaluna. She is brilliant. She's awesome. She is the assistant researcher to Dr. Jarvie, and Dr. Sue Jarvie does rat lung research at the College of Pharmacy. And I was their MBRE student for 3 years. Yay. Okay, this is a short outline. So my PowerPoint is mostly on like the introduction and background of rat lungworm, UVC, and X-ray irradiation. Then I'll go through the methods, results, and discussions. Okay, so rat lungworm disease. So neuroangiostrongyliasis, or commonly known as rat lungworm disease, is caused by the parasitic nematode Angiostrongylus cantonensis. So the picture, this first picture is of A cantonensis as L3 stage. So at this point, they are not visible to the naked eye, so you have to use a microscope. And then this middle picture is adult acantonensis. So you can actually see it's a small little nematode. And this was taken, dissected from a pulmonary artery of a rat. And okay, so it's called rat lungworm disease because the acantonensis larvae the egg hatches in the lungs of the rats, and then the larvae are regurgitated by the rats, and the rats will swallow the acantonensis larvae, and the larvae will go through the system and exit the rat through the feces as L1 stage larvae. And intermediate hosts like slugs and snails will get infected or pick up the acantonensis larvae through eating or coming into contact with the infected rat feces. And so when these intermediate hosts are infected, they could either infect paratinic hosts like like geckos and crabs, or it could infect accidental hosts, humans. We're accidental hosts because we stop once the larvae gets in our systems, we stop the life cycle of those larvae. So and the primary mode of transmission for humans is through consumption. So for us, eating acantonensis contaminated water, produce, or fomites um, is the most common way we get infected. And once we ingest the acantonensis larvae, they go into our stomach, and since they are parasitic nematodes, they are able to withstand the pH level of our stomach acid. They have this cuticle, whereas soil nematodes, if we ingest a soil nematode, they do not have this cuticle, and so they would die due to our stomach acid. And so if they're inside our stomach, they're stimulated by the acidity, and they will go through the intestinal lining and make it to our cerebral spinal fluid and swim up to our brain, where they can cause eosinophilic meningitis, which they burrow into the fatty tissue of our brain, which causes our body to produce excess amounts of eosinophils, and which can cause inflammation. And they can grow inside our brain. I think from L3 to L4 or L5 stage, and it's really rare to find adults in humans. Okay. Oh wait, okay, so here's an example. The bottom picture is the Parmeria martensi, a semi-slugs or intermediate host, and they are the most infectious slug species in Hawaii. So we use them for our studies because have the most load out of all of the slugs. Okay, and so research in Hawaii, especially the Big Island, is very important because Hawaii is the epicenter of in the United States. So Hawaii has the most recorded rat lungworm cases. And in 1933 was the first time a cantonensis was recorded in Canton, China. So it originated in Asia. And in 1960s, it was the first record of a cantonensis being in rats in Hawaii. And 1961, first man died in Hawaii. And prevention is very important because there currently is no cure for rat lungworm disease, but there are treatments. And if you, I think 2018, the rat lungworm task force in Oahu, they developed a preliminary guideline. So if you're interested, you can go to Hawaii State Department of Health and read more about it. Common symptoms, if you become infected, are nausea, vomiting, back pain, and in rare cases, you can go into a coma and it could be fatal. Yeah. Okay, and this is a graph of the number of rat lungworm cases in Hawaii from 1959 to 2019, and the red is Oahu cases, and then the green is Hawaii Island cases. So as you can see, it's Hawaii Island has the most number of cases throughout the years. And I checked in 2020, and there was only one case in Maui. A man is unfortunately hospitalized, I think in July. Yes, but that's good that there was only one case of rat lungworm. Okay, so electromagnetic spectrum. So yes, this is a graph of the electromagnetic spectrum. So my project involved in testing ultraviolet and X-ray irradiation on the rat lungworm, and UVC and X-rays are involved in, or on the ionizing radiation side of the spectrum. So ionizing meaning they can produce enough energy to break molecular bonds, cause DNA damage, as opposed to like microwaves and less harmful radiation. Okay, so why UV? Why UVC? So a lot of homes in Hawaii use rainwater catchment systems for their water. And here's a diagram. So when the water comes, it goes through a collection tank, then pump, then filtration, the filters, and then the UV or disinfection. You can attach a UV light. So my first paper was testing different size filters, but UV UVC lights are not required, so there's an option to have, like, some people don't even have filters or don't even have lights in their house, in their house, and which is kind of scary because on this bottom picture, this is inside a collection tank taken from a home in Puna, and as you can see, all of these Parmerian martenses are like gathered inside on the wall of the collection tank, which is, yeah, it's not good. Okay, so there's three different types, or there's four different types of UVC. There's UVA, UVB, and UVC radiation. UVA and UVB's rays we're exposed if we like go outside. We're most likely getting affected by UVA, UVB rays. UVC rays is the most harmful but is blocked by the Earth ozone layer. So if we go outside, we won't be damaged by the UV radiation. So we're testing the UVC because it's the most damaging. So there's have been studies showing the harmful effects or um, the comparison between UVA, UVB, UVC rays. So UVC is the most effectively absorbed by DNA. And there's two types of DNA, nuclear DNA and mitochondrial DNA, but both have been shown to absorb the UV rays. And UV radiation damage causes the formation of photodimers, which block transcription and elongation of transcripts, which could stop the production of like essential proteins. And then other study shows UVC activates pathogens, so they're unable to produce proteins, so which lead to the inhibition of cellular repair and growth, and they're unable to reproduce, which could lead to eventual apoptosis, the death of the pathogen. And this table is common organisms and bacteria that are found in Hawaii water systems and causes these diseases. And then on the right column is the UVC inactivation dose. So UVC is known to inactivate these pathogens. And then the units is millijoules per centimeter squared. So that's like adjusting the intensity of the UVC dose. And cellular repair of UVC damage. So nuclear DNA there's a process that or mechanism that nuclear DNA can repair itself, which is nuclear excision repair, if there is UVC radiation damage or a thymine dimer in this case is formed, the cell can detect it and physically cut it out and then replace the base pairs with DNA polymerase. So this is one mechanism. Yes, I think I deleted one slide, but mitochondrial DNA, they do not have this nucleotide excision repair. So because they don't have this repair mechanism, could cause the cell to inhibit its growth and reproduction. So it could eventually die. Because it couldn't repair the mitochondrial DNA. And so X-ray irradiation. So we wanted to test X-ray irradiation on the acanthenensis larvae because the primary effect of X-ray is damage to the DNA, just like the UVC, which is in previous studies is known to cause eventual death, inhibits growth and reproduction of nematode species. I think C. elegans. And then, so the USDA partnered with Dr. Jarvie at the College of Pharmacy because certain states were rejecting Hawaii local grown produce because they found intermediate hosts, the semi-slugs that could possibly be contaminated with acanthenensis in the produce. I think sweet potatoes, they found slugs in there, so they're rejecting our produce. And so the USDA, Dr. Peter Follett, he wanted to work with Dr. Jarvie to find at what dose of X-ray irradiation will have a huge effect on or prevent the spread of acanthenensis. And so currently the USDA uses 500 grays. Wait, no. Currently the USDA uses 400 grays as the universal insect phytosanitary irradiation dose. So they use it for like plants or other produce for insects like fruit flies. And in a previous study, they tested a cantonensis with 500 grays of X-ray irradiation, and they treated the acanthensis larvae with 500 grays, and then they fed it to mice and rats. And then if they found that at 500 grays, it wasn't infective to rats and mice. So the larvae didn't infect the rats. So they give the larvae to the rats and they dissect them after a period of time. And if they find the larvae anywhere other than, their digestive system, and if they're alive, then they will determine it infective, but they didn't find it in their brains or their hearts or their lungs. Oh, and this is just a picture of like a frog, a random frog. Okay, so my objective of both studies was to observe the mortality rates of the infective stage L3 acanthensis larvae after UVC and X-ray irradiation. Okay, so methods, you're finally to methods. So the first step is acanthensis isolation. So we physically have to go out and collect the intermediate host, the Parmenian martensi semislugs, because there is no current in vitro protocol for pure culture of acanthensis. So we can't just grow these larvae in the lab. We have to find the host and then isolate them. So the first step is to drown the slugs and then wait 3 days for the larvae to expel out of the body. And the third step, we take the bottom 10 milliliters of the test tubes, and then we use a microscope and we isolate the larvae based on the rat lungworm morphology and their behavior, because there are other microscopic soil nematodes or parasitic nematodes. And then here's a picture under a compound microscope, and then this is a video, so hopefully it works. If you can see them wiggle, yeah, some of them are active, some of them are wiggly, yeah. Okay, and so treatment. So for UVC radiation, we used a UVC crosslinker at 254 nanometers. And here's a picture of the crosslinker. It looks like a microwave. And for each dose, so on the right side, here is all of the energy or the UVC dose. For each of these doses, we did 5 replicates of 200 larvae. So we did like 1,000 larvae per dose. And so 0 millijoules per centimeter square is actually the live control because it has no treatment. And then we did a dead control. And so we did the 0 millijoules, so the live control at 30 degrees Celsius, because we noticed towards the end, towards the higher UVC doses, that the petri dish was hot getting out of the crosslinker. So we wanted to test at a higher temperature for the live control. For reference, the room temperature is about 20 degrees Celsius. Yeah, and so the highest was 5,000 millijoules, and then the lowest was 25. And we wanted to test above 40, so we tested 25 and 50 millijoules per centimeter square, because 40 is the National Sanitation Foundation domestic rainwater catchment range. So it's the national energy dose for the water catchment system. Okay, for the X-ray irradiation, we used a linear particle accelerator. So on the right, here's the picture of the machine, and the energy dose was 200 to 1,000, and we did a live and dead control. and then 5 replicates of 200, so 1,000 larvae per energy dose. And we put the larvae into these test tubes for each replicate. And yes, the bottom right picture is one replicate. And here's a picture of a linear particle accelerator. I was pretty excited to find this moving picture. But over here you have your ion source, and then this machine accelerates the ion charge at a high enough speed to reach that the energy dose, or that graze. And then over here is where, okay, so the ion charges and then it moves along the linear beam and then to the target. So right here would be all the tubes of the larvae. Okay, so data collection. So once for the UVC and X-ray, once they're both treated, we put them into separate plates. Here's a picture of the plates. I believe this is like 384 wells. And then we put it into this machine, which is the Operetta high content imaging system. So it's like a huge microscope with a camera or recorder. Yes, and I use these 3 filters to determine mortality. So the first filter is bright field. I use it to determine if the image is larvae or debris. And then I use fluorescein and propydium iodine to determine if they're dead larvae. So propydium iodine, we used 0.25% stain. So propydium iodine, it binds to DNA. So it can't get through the cell wall or the membrane of the cells. So the cell has to be lysed open, so the cell has to be dead in order for the propydium iodine to bind to the DNA and for it to fluoresce. So only the fluoresced larvae we can determine dead, because the cells would be lysed open. So this is a picture of a dead control. So we expected it to fluoresce, bright. Yeah. So here on the right, this is a picture of the filters fluorescein and propydium iodine. So as you can see, I would determine these bright larvae as reach complete mortality because they're fully fluoresced, as opposed to this green larvae, which did not pick up any propydium iodine. Yeah, and I wouldn't count this one. That's kind of yellow. Yeah. Okay, so next is genetic and statistical analysis. So we wanted to determine that the larvae we were testing were in fact acanthenensis. So we had to do DNA extraction. So we took larvae from, I think this is a picture of the UVC radiation experiment, and we extracted the DNA, and then we did a real-time PCR. And this is a picture. So the if the DNA was in fact acanthenensis, it would fluoresce above this threshold line, and each color is a different replicate. So all of our replicates fluoresced above the threshold line. And so all of our replicates for the UVC study had acanthenensis. Okay, and then we did statistical analysis on our results. So we checked for normality, equal variance. Yes, okay. So results. Okay, so UVC results. So, no, okay. So the top line, this blue line, is the dead control. So the x-axis is the time after irradiation, so the days, and then the y-axis is the average percent mortality. So the dead control is this top line. So the dead controls were 100% stained, so they're 100% dead, if that makes sense. And then the live control is actually I think it's this one. This one is a live control. Wait, can I move this? Okay, yes. Okay, yes. This navy one is the live control, and then this the this one is the live control at 30 degrees Celsius. So as you can see with the increase in UVC dose, the faster mortality, but the faster the larvae reached mortality. So the highest dose was 5,000 millijoules, and that reached mortality at day 3. You can't really see it, but it was this top line, so it reached 100%. And then 25, 50, and 100 millijoules centimeters squared is right here, and they were very similar, and they were like the lower energy doses. Okay, and so we tested the live control at 30 degrees centimeters because the temperature seemed hotter. And here's just a graph of this darker blue is the temperature of each UVC dose before exposure. And then the light blue is the temperature after the UVC. So the temperature of the petri dish that we put the larvae on, and as it increased at the 3,000 and 4,000, 5,000, you could see that there is temperature difference. Okay, so results. So yes, the faster mortality rates occurred with the increased UVC dose, and the 5,000 millijoules centimeter square treatment reached complete mortality. Day 3, the lowest reached 70% by day 30. Yes. Okay. Yes. And the 30 the live control at 30 degrees didn't reach more, or they didn't die as fast as the normal live control at room temperature. And then genetic and statistical analysis is ongoing. Okay, so for the X-ray irradiation results, so the x-axis and the y-axis are the same. The x-axis is the days, and the y-axis is the mortality rate, so the percent of stained larvae. And as you can see, it doesn't seem to have a dose response. So the dead control is this top line, and the live control is somewhere in there. So we wanted to zoom in. So here's a graph of the highest is 10% mortality rate. And as you can see, this top line with more mortality percentage is 800 grays, which is weird because this 1,000 grays is like in between, like right next to the live control. So for the results, there wasn't more than 3.3% mortality for the X-ray irradiation. And there's no dose response after 23 days among all of the energy dose. And there was a significant difference in percent stained larvae found among the irradiated larvae versus the live control. And then the 800 grade had a slightly elevated mortality. Okay, and so since there wasn't any response, we wanted to check if we could physically see if they were alive. So we did an acid test on day 26 with the X-ray treated larvae. So the x-axis is the treatment, and then the y-axis is the number of moving larvae, so the larvae that we physically saw moving. So this blue bar is the larvae just in water, so kind of like our control. So we counted the number of larvae that were moving just in water, and then we added 0.5% pepsin and 0.7% HCl to mimic the stomach acid, because the stomach acid is known to like stimulate the acanthenensis larvae. And so as you can see, there was a lot more movement. There was about 25% of the larvae moving, or 25% more of the larvae moving than the larvae in just water. But there was no statistical difference found between the treatments of number of moving water in the water, or there was no statistical difference between the treatments in the number of acid, pepsin, and HCl mixture. Okay, so discussion. So UVC. So the high mortality rates in the high UVC dose could be because of the prolific DNA lesions beyond the cell's repair. So they could be dying with the higher doses because the cells can't repair themselves. Or so the low mortality rates in the low dose UVC treatments could possibly be because they are able to repair the nuclear DNA with the nucleotide excision repair mechanism, and then they could repair some mitochondrial lesions from the UVC damage. So it is unknown of the viability and infectivity of the unstained larvae. So with the stained larvae, we can determine that those larvae are or reached complete mortality. So the unstained larvae are dead, but we cannot say that the stained larvae are dead, but the stained larvae are dead, but the unstained larvae, we're not sure, that we cannot say that they're alive because we don't know if they are alive or infective of rats or humans. So it is unknown effective of the water catchment UVC system in preventing neuroangiostrongylosis. And then while it is unknown why the 30 degrees Celsius non-irradiated larvae had lower mortality rate than just the live control in the room temperature water. And then the temperature from prolonged UVC exposure did not affect the mortality rate in the high UVC treatment. And for X-ray, so although our studies show X-ray irradiation doesn't cause death, we suspect that X-ray irradiation caused some sort of or inhibits larval growth and reproduction, because there was a previous study done on acanthenensis treated at 500 grays, and they turned out not to be infective to rats. And it is unknown why the 800 gray treated larvae had a statistically different mortalities than 1,000 grays. And then the physiological state of the unstained non-mobile larvae of the X-ray irradiation is unknown. So the unstained larvae and the non-mobile larvae, we don't know if they're alive. Yes. Okay. So future directions. So higher UVC and X-ray irradiation doses should be tested to find a more immediate death and a more efficient way of disinfection. And then testing of household water catchment disinfection models is needed. Yes, we need to increase the energy dose to find a faster mortality rate. And then we should also explore other options of water catchment disinfection. And then a rat study is needed for both studies, UVC and X-ray, to determine if the unstayed larvae are infective to rats. And X-ray irradiation of semi-slug reproduction. So we should irradiate semi-slugs, or find that the optimal X-ray dose to prevent the spread of rat lungworm, if the semi-slugs are found in exported produce from Hawaii. Oh, and just for fun, this is what my job was at the USDA. I was actually hired as a entomology lab assistant, but my main job was working with slugs. Um, here's a picture of the adult slugs, and uh, we had to collect the eggs, and here's the close-up of the eggs. It looks like fish eggs, like the salmon eggs, just like clear salmon eggs. And then here's a picture of the eggs underneath the compound microscope, and you can see the little slugs developing. And then here's also a picture of them hatching, and then for reference, here's my glove, and so they're really tiny. And it's kind of scary if you think about it, if like they're infected at that small age and then found in like lettuce. Yes. And here's a blown up zoomed in picture. I think they're kind of cute, but I've been staring at them way too long. Oh, and also fun fact, this is a picture of a slug repelling. So I didn't know this before I worked in this lab, that slugs repel. So if they're on a branch, they could use their slime, they can expel slime, and then like Spider-Man, like crawl all the way down to the floor. And I think this is a image of a slug that like turned around because his butt is like right here, and then his head is like crawling back up. Yeah. Okay. So acknowledgement. So I worked under Lisa Kaluna and Dr. Jarvie at the College of Pharmacy, and which was also funded by the Hawaii State Legislature. And I worked under Lindsay Hamilton and Dr. Follett at the USDA. And then my project was funded by the NIH through IMBRI. And yeah. Oh, yep, here's my references, and thank you. Um, yeah, if you guys are interested in IMBRI, like I highly recommend you contact Dr. Jarvie or about IMBRI. Here's the IMBRI email. Yeah. Oh, and if you have any questions about like my research, you can email me. Yay.
Thank you so much, Yaeko.
Yeah, thank you. Thank you for having. Good luck, guys. You're almost there. Yeah.
Thank you. Does anyone have any questions for Yaeko?
If nobody has any, I can start off with one.
Okay.
I'm like extremely interested in getting into research right now, and I'm so sad that I got interested in it in my like 5th year, not even senior year, but like after senior year. Yeah, so right now I'm involved in a little research too. It's with water quality in Hawaii, but I was gonna ask, what was your experience involved in like a publication, because that's something that I want to look into also. But did you like have like a lot of responsibility to look for references and write up a background or compiling data, or what was your
yeah, my main job was like working with the methods, because that's what I did. I mean, I helped a little with UV research, like reading different papers, but we haven't published that yet. My first publication was about the water catchment systems, and my main job was like just collecting the larvae and like putting in hours, like isolating the larvae. So I was mainly there to like gather the larvae for my first year, because I didn't have that much experience. But the second publication that took off when I was hired for the USDA and we were rearing the slugs. Um, but I mean, I think even if you're in your fifth year, like you should try and or wait, are you in like a research?
Well, sort of, but not like a publication, I don't think.
Okay.
I'm like more of a volunteer. So I was wondering,
Okay.
with you being involved in a publication, did you have an active role in like writing it or
Oh,
something?
writing. Oh, just the methods. Yeah,
Okay.
just the methods, because that's what I was doing. But when I started, I was just like I didn't like expect to be in a publication. I was just like, okay, what do you want me to do? I'm willing to, like just be a part of like anything that involves research. So I was just like excited, and I think I got like got so much experience and got so much publications because I was so open in like
Mhm.
doing anything. Yeah, so yeah. Yes.
Nice. Thank you.
Yaeko, could I ask you to unscreen
Oh,
share so then
oh
like we can
yes.
all see each other?
Okay.
But I
Okay.
loved your presentation. Thank
Oh,
you.
thank
I have a
you. Thank
question,
you.
Yaeko.
Oh yes. Yes,
Question.
I'm listening.
Okay, so did you ever look at Sharp or did you just go straight into
Sharp.
INBRI? And why did you go straight into INBRI if you only
Oh,
looked at INBRI?
yeah. I applied to Embry my sophomore year. I didn't know about Embry or Sharp. My friend Bruce, he was Lisa's first or one of Lisa's first Embry students, and he was my friend. And so he was like, "Hey, do you have any research experience?" And I was like, "No." And so he was like, "Hey, come to our lab." And so that's how I got sucked in. Yeah, I was actually wanted to just do like a semester of Embry, but then I fell in love with it, so I just finished like my whole college experience. Yeah, yeah, but I'm
So,
not sure about Sharp.
to sort of answer your question, Alyssa, I've looked into both Sharp and Inbree. I personally don't really like research as much as Yaeko does, so I'm not super passionate about it.
Okay,
But Sharp differs from Inbree in the way that they structure it, sort of, and pay and everything like that. So Sharp treats it more as an actual job, while Inbree, I know that there's like a certain amount of hours, I think, but you get a stipend at the end. So Sharp, in that term, you could get paid more or less than Inbree depending how many hours you put in. I don't know how, both of them are running it because of COVID and everything. My lab advisor was also a Sharp advisor, so she was telling me that these symposiums are different. So where Yaeko had said earlier that you get to go to JABSOM with INBRI, and by the way, if you are in INBRI and you go to JABSOM, that is paid for. So you get a tour of JABSOM, the John A. Burns School of Medicine. You'll basically get like you spend a day there and do the symposium. The symposium that Sharp does is not at that same level. It's like not as professional. It's not as intimidating as what they told me. It's more like all of the advisors and lab people for Sharp kind of just do a symposium at the pharmacy school, I think, and you present it to like other lab advisors and that kind of stuff. Whereas Inbree, I think, was open to the public. Maybe I never
I
went
think.
to one of the symposiums. Also Sharp is kind of more for the like disadvantaged groups. So if you're like a minority group, like a native Hawaiian descent or that kind of stuff, or like Latino, that kind of thing, you get not only priority, but that's kind of how you would get in, while Inbree kind of accepts every undergraduate student. And I know like UH's one last year had like a lot of openings for research undergraduate research assistants. Sharp also yeah, so like under, like I was saying, the different descents, but also if you receive like the Pell Grant and stuff, that's how you would do that and actually qualify for Sharp. Their projects are very similar. I think you can also be published from Sharp as well. And Sharp's kind of main goal is to create biomedical researchers. So if that's not like what your goal is, it might be better to go with INBRE, because I've been told by like my old lab advisor that technically if you're not interested in biomedical research and that's like not your end goal, and your end goal is like more to be a doctor, they would consider you like a failure, because that's their whole thing. Like they want to create biomedical researchers. That's like the whole thing with them. So it's different in those perspectives. They're very similar though. So I'll just leave that up to you. But yeah, that's what I've been told. There might be more, might be less, but yeah, that's
Thanks so much, Victoria. That was actually super helpful. Is there any other questions?
Yeah, I do have another one.
Okay.
So I looked at the INBRE website and tried to apply for it. At one point, it was like open for application, but I guess I missed the deadline. It doesn't say when the next application cycle is. Do you happen to have that information?
Yeah, it's in spring, but I think you should try and email them now, because when I started in October and like I missed the date in like I think it was August or September. So yeah, you should try just ask them, just shoot them an email, I think at embry at hawaii.edu.
or Dr. Jarvie,
Yeah,
who is
Dr. Garvey.
Yaeko's lab advisor. She's
Yeah.
also the campus coordinator for Hilo. And I remember before, like last semester, they were still accepting applications. Like technically, you would just have to do it through Dr. Jarvie, and she would like turn it in for you, because I know Hilo had a lot of undergraduate openings, and a lot of them are like with the pharmacy school. So if you're able to, and you're like, oh damn, I missed the deadline, I don't know how this semester, like I didn't apply, I probably won't be doing research again, but um you could um you could just email Dr. Jarvie and ask if they're still accepting applications, because they might.
Oh yeah, and there's like other different labs. So if you're not interested in rat lungworm, there's Dr. Ingo. He does neuroblastoma research, so cancer research, and then Dr. um what was your Victoria, what was your what was your project?
uh I I don't know what my project was. My lab advisor, or my old lab advisor, Dr. Tan, she is a pharmacy professor as well. And she did a lot of pharmaceutical research. She did hers with cancer cells as well, but she does a lot of like cellular work. So, like, what we're learning kind of in cell bio, it's pretty similar to what she does. So, if that's interesting to you, maybe she would be a good fit for you. But also, like, if you have no idea, you could also ask Dr. Jarvie, and she can set you up with somebody who might match your interests.
I also wanted to add something to that. So I've talked to like dozens of people in medical school, everyone, who gives advice to pre-meds. And there's kind of been like a resounding answer to the question of research. That is, it kind of doesn't matter to some degree what research you get involved in. So, a lot of research, a lot of, like programs like, like cell bio and all these different kinds of things that have to pertain to like microbiology and kind of like doctory medical things. While they are great, they are usually pretty competitive. So, if that is an issue, any sort of research at all is actually super helpful. And honestly, the biggest asset that I think that medical schools are looking for when they're looking at research is just not necessarily like, like what you're exactly researching, because, that could be obsolete in a year, you have no idea. So I think that what important parts of research you know, a lot of medical schools are looking for is just that you know how to do it, you're professional, you know, you can stick to something, you know, like longevity of research I think is really important. You know, if you're staying with something for, you know, more than a year or something, I think that, you know, that's a really good indication to med schools, you know, like commitment to research and all of that sort of thing. So like I know like I took like one research course or something, like a directed study or something with Dr. Muir. He's like a biology, like an ecological biology professor, and he like it kind of doesn't matter. Like it doesn't matter what kind of research you want to go into. It doesn't have to be, you know, super medical. Just because, you know, just simply like knowing what an IRB is, knowing, you know, the scientific method in and out, you know, how to build references, you know, like proper lab technique and all of that kind of thing. Like those are skills that you can put on your resume, and that, you know, those do stand out as far as like the facets of research that are really important to, like, you know, getting into medical school. So I think that, yeah, you know, like Yaeko is kind of a golden star as far as, you know, what med schools look for in research experience. But honestly, like I think any sort of avenue of research is kind of like thumbs up, you know what I mean? So it's not so much of a pressure, I think, to like get into, like, you shouldn't feel a ton of pressure to be, you know, super involved with like INBRI or something like that. Any sort of avenue of research I think is like helpful. Yeah. Was there any other questions for Yaeko? Sorry.
I have a question.
Okay.
But this is more for everyone. For nursing students, can we participate in research too?
Yeah. My lab instructor, Lisa Kaluna, she said her first embry students were mostly pre-nursing. Yeah, they just wanted to get biomedical research experience. And so yeah.
But is it required for nursing
Oh,
school?
I'm not sure for nursing, sorry.
For nursing school, I don't necessarily think it's required. It does look good if you want to be competitive, because when you go into it, you're going to look similar to everybody else. So if you are able to say, I'm different from this other person because I have research and I'm putting myself out there and doing more things and becoming a well-rounded student, that shows a lot to that shows a lot of people who are going to be judging you. And from what I understand, it's not required, but it will look good for you if you decide to go into nursing school.
Can we participate in research as a freshman, or is it from upperclassmen?
I think it's like 2 semesters. I think you have to check the Embry. Well, for Embry, I'm not sure about Sharps, but I think you have to check the website.
Okay, thank you.
Most programs, you have to be at least a sophomore or have sophomore status and credit wise. But to volunteer in a lab, you can probably just ask professors.
Mhm.
Thank you.
And I also think for age, you know, INBRI and like Sharp, those are super great avenues because, you know, they are like localized to what you may need, you know, as an undergraduate student. So those are super helpful resources. But I mean, being proactive, you know, at any age is like a super idea because, you know, you could, like, for example, I'm a second year this year. I'm only a sophomore, and my freshman year, I got involved with that, you know, research directed study with Dr. Muir. And, you know, it's just kind of a thing where I don't think that there's any age limit to ambition. You know what I mean? So if you want to go out and do that, I think, you know, more power to you. Yeah, but as far as nursing goes, I think that I think the best answer to that is kind of a question of where do you stand as far as, you know, like time, and then also like you know, like the efficacy of, you know, like how you commit to things. just because, you know, research is a big thing. And while that could be helpful, I wouldn't put all your eggs in one basket of, okay, I'm going to focus on research, but, you know, my, you know, grades may falter or something like that. You know what I mean? So I think that it's kind of a thing of, if you have free time, then absolutely. I think it could also open windows to, you know, if you want to go further than nursing, you know, because you're in a pre-med club and you're in nursing, like that shows ambition enough. And I think that maybe that could translate in the future to, you know, something beyond nursing. So I think that if you do have research, that opens a lot of doors for you that, you know, not all pre-nursing are going to have. So yeah, I think that would be like the best answer to your question. It is, what's the time in, is it 5 o'clock there? Sorry, I'm 3 hours ahead. Yeah,
Yes.
it is the end of our meeting. But if anyone had any more questions, feel free to email the AMSA email, and then we could also like rebound those questions to Yaeko. And then also I can stay, and I'm sure some other people can stay a little bit longer if you have any other questions. But as of now, I think it would be okay to end the meeting recording.
Okay.